Okay, good afternoon everyone, and welcome to our Strategic Cereal Farm Results webinar on farming and the environment. My name is Anna Reynolds and I'll be your host for today. Today we'll be covering cover crop trials across strategic farms Scotland, East and South, as well as our flowering strip trials. We'll be examining effects on biodiversity, water quality and soil health. Pretty much everyone, as you can see, involved in the trials is here today, so plenty of opportunity to have your questions answered. Some general housekeeping points from me. Everyone except for the speakers is on mute and we can't see you. If you have any questions for the speakers, or if you're having technical issues, there's a box in the right-hand side of the screen where you can type messages to us. Only speakers and AHDB staff can see them, so they're private. If you didn't enter your base or Narosa details when you registered, or if you're not sure, please write your name, membership number and postcode into the question box so we can register your attendance to get points. As with the questions, only staff and speakers are able to see this. This webinar is being recorded and will be available to watch later, and we're scheduled to be here until 2 o'clock. So without further ado, let's start by looking at baselining, and we will begin with soil health. So if I can pass it over to you, Callum. Thank you.
Hi everyone. So I'll just very quickly introduce myself and the work that we're doing at Strategic Farm South. So I'm Callum Scottson. I work for NIAB. I'm based in the South region at the Sutton Scotland office in Hampshire. I am effectively, so my background is in soil science and looking at where plant science meets soil science in terms of how roots interact with soil and that sort of thing. And I manage an amount of the sort of soil and systems projects in this region, including the Strategic Farm South, based at David Miller's in sort of near Basingstoke in Hampshire. So if I can have the next slide, please. So one of the 4 projects that we've got at the Strategic Farm South is looking at farm scale soil health baselining. So the idea is that we're assessing soil health in ways that are feasible and sort of possible to undertake at farm scale across whole farm systems, basically. So that means it's fairly stripped back compared to some of the other ones because it needs to be feasible to do at that scale and not be too demanding in terms of time or resources. So in terms of the project that we've got at Strategic Farm South, it involves 4 fields. So it's looking at these 4 fields at that broader scale. All 4 of them have wheats, 2 are spelt wheats, 2 are sort of standard winter wheat varieties. And effectively, it's that idea of that long-term monitoring. So effectively, we're trying to build up some farm scale benchmarks for soil health, look at how we can implement some existing systems for monitoring soil health, collate that spatial data to try and work out where we can actually do that soil sampling to put some data to soil health, and completing various assessments and things like soil health scorecards, which we'll touch on in some more detail in a moment, and things like monitoring the crop development and disease as that runs in parallel to the soil health assessing. And then finally also looking at the yield as well. That's obviously one of the most important elements for most of you. Next slide, please. One of the first things that we do is we look at establishment. So this gives us a really nice idea of, are the crops that we've put into those fields establishing well? Are we going to be able to get nice representative results? Obviously, if a crop hasn't established in a field for whatever reason, you would expect that to have knock-on implications in terms of soil health and all manner of assessments. So really, it's about gauging that there is sort of fair comparisons that you can make, and that it's a decent decent crop that you can assess to then come back to in future and make those fair comparisons in time. So effectively, we're checking up on the establishment and the uniformity of establishment, making sure it's fairly even across the site, and making sure that there's no problem areas. And if so, we can sort of either look at those in more detail, or if we know why there's a reason why a particular corner of a field might be anomalous, we can sort of exclude that from some of the broader comparisons that we make field to field. So in this instance, for this project, for the 4 fields we look at here, demonstrated that establishment was successful, and by week 8 post drilling, it was pretty uniform across all sites, which is good. Next slide, please. So we've had that initial look at establishment. We know that all of the crops are doing reasonably well early in the season. So what do we do next? Well, the next thing we do is soil health scorecard. So this gives us that nice baselining to our soils, and it gives us a nice sort of standardized system to use. So it's a system that was developed by the Soil Biology and Soil Health Partnership, which was led by AHDB and BBRO, and there was a whole load of other sort of stakeholder organisations that fed into that to develop this system. And it basically provides a traffic light system for assessing soil health matrix. metrics. So the idea is it ties together chemical, physical, and biological features of soil, gives them a red, amber, or green color coding, depending upon how they performed. So if something is outside of what would be considered a typical range for that particular metric, it will be red. That means that you need to monitor it more intensively, you need to go back and monitor it again sooner than you might otherwise. If it's amber, it means that you probably want to keep an eye on it and actually look into what's taking place, but it's not sort of an immediate concern. And if it's green, it means that it's probably within that acceptable range and you can check it less frequently. It doesn't need such intensive monitoring. It does also adapt for things like whether your soil is light, medium, or heavy in terms of soil texture. So this example on the right-hand side for organic matter percentage, it does give you some sort of some flexibility within features such as that to change for soil texture. And really, it's about monitoring those changes through the rotation. So you can check it every other year, for example, as we do for this project, and it will give you that idea of change over time. So you effectively choose some sites within the field, geolocate them using something like what 3 words. We normally choose at least sort of 3 sampling sites, and then sample within a 5-meter area within a radius at each of those sampling sites. Do some in-field scoring, some sample collection, and send those off for analysis. So the idea is that it ties together a VES assessment, so that visual evaluation of soil structure, worm counts on that soil block that you've extracted for the VES, and also you send off a sample for lab analysis as well. So that's where you tie together that chemical, biological, and physical data. Next slide, please. So to use an example from this project that we've done here, so this is the sort of summary page of the soil health scorecard with that color coding that I mentioned, and it enables you to pick out some things immediately. So for example, the VES assessment, which again we'll come into in a little while, that VES assessment in Big Grange, the soil structure was noticeably that little bit tighter, so it flags it up as amber, something that needs a bit more investigating. The important thing to remember with the soil health scorecard is that you have to sort of put some of the numbers into context where you know your fields in terms of, for example, the pH here. It's on a sort of Hampshire chalk soil, so it's going to have a higher pH, hence it's amber coloured. There's not a huge amount you can do about that. It's just a feature that you would know about your soil, so you'd see that amber colour and think, okay, well, actually, that's easily explained. Another figure that's sort of pulled out here is the red colour for the pea in piggery. And I mean, because of the name piggery, that would suggest there's probably a history of livestock there. So again, it's one of those things where it's not necessarily something that requires immediate action, but it does require that bit of thinking to think, okay, what's the sort of reasoning and the sort of source behind this value that I'm getting. Next slide, please. So as I mentioned, one of the core components of that soil health scorecard is a VES assessment. I'm sure you've all come across these by now. Basically, it's a simple method for assessing soil structure, requires only a spade. The idea of all of these assessments that we do here requires very simple kit, a soil auger and a spade essentially, and some guides that are free online. You effectively extract a block of soil and you give it a score of 1 to 5, 1 being very well structured, 5 being very poorly structured. And you assess things such as the size, shape, and firmness of aggregates, pore distribution, and things like the soil colour and smell as well. Guides for this can be found online, including on the AHDB website. There's loads of good resources for this, and it's a sort of fairly straightforward procedure. But that's how we look at soil structure. Next slide, please. Ooh, next slide. There we go. Thank you. Ooh. There we go, wonderful. Thanks very much. So that's one. In terms of then, we look at some examples. We'll look at some examples here of the VES assessment from this project. So again, it's a bit of that case of looking at that amber color and thinking, okay, well, what was going on there? When you do a VES assessment, really good idea to take photos. You can take photos both of the block once you've extracted it and of some smaller bits as well. Here on the left-hand side, you can see that comparison of Big Grange with that tighter soil structure, larger aggregates and a more rigid shape, versus Piggery and Ashen Grove, which had those much smaller aggregates, really nice friable structure. Waltham Marks on the right then sits somewhere in between, where there's a few larger aggregates, not quite that nice sort of small stable aggregate structure of Piggery and Ashen Grove, but a little bit tighter. Next slide, please. So then once we've looked at these features of soil health, we then tie this all together and look at yield at the end of the year. We also do some disease assessments. So the disease assessments are very straightforward. Again, guides online. That's just a case of what you or your agronomist might go out and do, just assessing your standard cereal diseases. We normally give them a percentage score for the purposes of things like research and tracing it, in terms of percentage area of the leaf covered. And we just monitor that throughout the year. In terms of this project, very low disease levels that year, so there wasn't all that much to report. Low levels of septoria, but that was about it. Then we look at the yield. So this is just taking those yield values that come off the combine, and comparing those between the fields, looking at them longitudinally as well, just making sure that actually they're keeping pace with one another. No significant differences in average yields for this year. So both of the spelt wheat crops, so that in Ashen Grove and in Big Grange, no significant differences there, both at about 8 tonnes a hectare. And in terms of the winter wheat in Waltham, Marks and Piggery, those two, no significant differences between those sort of conventional winter wheats either. So it just gives you an idea that those fields performed relatively well throughout the year, and actually Big Grange, despite having that tighter soil structure, actually still produced just as good a yield as Ashen Grove. So it's one of those things we then keep an eye on it and monitor and see that change over time. Next slide, please. So in terms of some take-home messages from this, really the idea is that we emulate the format and the intensity of soil health monitoring that's feasible at the farm scale. So we don't use any specialist equipment or anything like that. All of the analysis is performed by standard labs, sort of commercial laboratories. The idea is that it's also done on the sort of time scale that would feasibly be done on farm. So these fields are going to be reassessed in 2024 or harvest 2024. So we get that every other year sort of snapshot of what's taking place. Because soil transforms fairly slowly and most changes in soil take place fairly slowly, every other year should be good, and we should be able to demonstrate that that's enough to capture those sort of macro changes in soil structure and in soil metrics, basically. And so, yeah, the idea is that we come back in harvest 2024, take another look at these fields and see that change over time, and come back and refer back to how they've changed between these two sets of harvests, really. And that's it for my slides. So I'll pass over to Nathan Morris. He's going to talk about baselining for water quality.
Okay, good afternoon, everybody. I'm Nathan Morris, and I work at NIAB as the Farming Systems and Soil Specialist, and I'm involved in a range of projects around farming systems and soil management. And today we'll just talk a little bit about the water quality and how we might look at baselining and measuring that on farm. Next slide, please. So in terms of baselining water quality, we are obviously all aware that water quality is a very important part of our environment, and the environmental costs associated both in terms of the aquatic life and also financially, and the treatment, particularly for drinking water, and the costs associated with that have been estimated at up to about £1.3 billion per annum. There are support programs through, for example, the Catchment Sensitive Farming Initiative, where growers can seek advice and also obtain grants to help mitigate against water quality and water pollution. There are also other support programs, particularly from water companies that may have catchments that are more at risk from leaching, and those particularly help support growers in perhaps using cover crops as a way of mitigating against that risk of leaching into the watercourses. Next slide, please. So, as I mentioned, in terms of trying to mitigate against diffuse pollution, there are quite a number of tools that farmers have available, particularly looking at some of the on-farm management in terms of things like cultivation practices, maybe introducing cover crops on farm, looking at their soil management programs, looking at land management in general, particularly around nutrient use, slurry storage and manure management, but also water resources and obviously the flood mitigation measures as well. So there's a number of different approaches there that, as I mentioned, we can get support to help look at and improve to minimise the risk to leaching. And just as an example here, there was a DEFRA funded project as part of the demonstration test catchment. This is results from the River Wensum in Norfolk, where they were basically looking at various mitigation measures that were around cultivations and the use of cover crops. And there was very good data to show the value of cover crops in reducing that risk of leaching, nitrate leaching, by around 80% to watercourses. And obviously cover crops and particular types and species of cover crops can be very good at deep rooting and obviously having the ability to scavenge nutrients deep in the soil profile and obviously reduce that risk of those nitrates being lost to water. Okay, next slide, please. I think that might be it. One more, I think. Yeah, so in terms of the strategic farm in the east that I've been working with, we've been looking at water samples collected when the field drains are running, which again is driven by rainfall, and then obviously getting those analysed. This is through Essex and Suffolk Water to look at known both pesticides and nutrients in that water. But as farmers, we can also take water samples from field drains if we feel that there are or is a risk from pollution, and send those to commercial laboratories as well for either a suite of analyses or specific. So things like nitrates can be quite easily and at relatively low cost analysed by the lab, and you can have a look at to see if there's an impact from, you know, maybe a application of manure that or the storage of manure that may have risked some leaching to the watercourses. And as I'll go through in a little while today, we will consider the effect of cropping and tillage approaches and nutrient applications on how that might risk the sort of increases in nitrates in water quality. So thank you very much, and I will pass over to Aoife and Patrick.
Good morning. So hi everyone, my name's Aoife O'Driscoll, and I think Patrick should be on the line as well if he wants to turn his camera on or hang on. So yeah, we're going to talk in the next couple of slides about the flowering strips work that we've done at Brian and Patrick's farm. And if we could just move to the next slide, please. So at the farm, we basically installed flowering strips in 2 fields and had 1 field with no flowering strip. And what we wanted to do was, we had one kind of overarching question, which was, can flowering strips affect the diversity, distribution and number of pests and beneficials in an arable system? And to answer that main overarching question, we had 5 kind of smaller sub-questions. So, and this dictated how we did our baselining. So the first one was looking at what we call flowering phenology, and that's just basically looking at when flowers are in flower across space and time, so within and across fields. We were also interested in weed abundance. So did the plants in the margin encroach into the main crop and cause weed pressure? And then in terms of pests, so we were focused in this project on slugs and aphids. So were there different numbers of slugs and aphids at the beginning and then over time within and across the 3 fields? From beneficials, so then how did the flowering margins impact on their numbers? Again, both within the field, in the margin itself and then across fields. And then I think most importantly was how farmer friendly are the monitoring methods that we used? So can they be put into practice by everyone, no matter what your level of skill set is? So next slide, please. Okay, so this is a layout of the 3 fields. So at the top left square there, you have the 3 fields. So the farm standard with no flowering margin around the outside. And then we had a second field with the margin the whole way around, and then a margin in the center of the field. And then a third field with just a margin around the outside. And what we did to do our kind of baseline monitoring was we went out with a GPS, so a little iPad with a GPS attached to it. And because we were doing this over multiple years, we GPSed the points while we were out there. So across two sides, around the top side and the right-hand side, we had 3 monitoring points, which were around 50 meters apart. And we GPS those so that we go back to the same location each year, and to monitor the specific effects across space and time. So the points were within the margin itself, and then 100 meters into the field, to look at, do you get spillover of insects or plant species into the main crop. And then we also had a monitoring point 50 meters from the center of the middle margin as well. So those points were GPSed, and we could go back to the same points each year. But again, you don't have to do that. You could just put a stake up at those specific points if you were interested in doing this, and monitor those points over years to look at change over time. Okay, so last slide for me. So how did we do our monitoring? And this was something that was done right from the beginning of drilling all the way through to kind of late June, early July. And we did various things to look at plant species, pests and beneficials, and also solitary bees as well. So we started off in autumn, winter, kind of October time, and we put out slug traps and pitfall traps. Now, the slug traps were basically just plant pot saucers put down at each of our monitoring points with a little bit of muesli, and left out there for 3 days just to look at slug numbers across the fields and within the margin and the field itself. And then we also had the pitfall traps, which are just like a plastic food pot that you dig into the ground, fill with a saline solution with some washing up liquid, and again left out there for 3-5 days. In the wintertime you can leave them out for a bit longer because there's less insects about and it's colder. So you can leave them there for up to a week and they would be fine. So that was kind of October, November time. Then in spring and summer we wanted to look at again changes in number of species across the year. So we also used pitfall traps, and we used these aerial water traps. So again, they're just a big plastic pot that we secured off the ground. And the idea for those water traps was you were picking insects that were on the crop, but are on the margins or in the crop itself, and that were falling into the water. So any, say, beneficial like ladybirds or hoverfly larvae that were living, say, on the wheat crop, that they would just fall into the water and you would catch them that way. And you wouldn't really do that with a pitfall trap. The pitfall traps are more for ground dwelling insects. So we had pitfall traps, water traps to do our insect monitoring, and then we also went out and used quadrats just to look at plant species at a single time point. So we put 9 quadrats and 5 meter spacings across the margin to look at the variation in flowering grass species across each of the margins. So again, very, very simple things you can do to look at plant and insect species. And then the final thing we did was look at solitary bees. And again, this was a very simple experiment that actually Laura Nicole at SRUC came up with. So all it is just a piece of gutter pipe, and you cut it to size and you cap it at the end. And you can buy solitary bee nests from the internet. There's lots of different websites you can buy them from, and just slotted them in and stuck them on a pipe and put them out kind of from March until August, and you collect them back in and you look at whether there's any solitary bees in them. So again, really simple thing you can do, and there's nothing here that is kind of beyond the skill sets of anyone to look at. So that's that slide, and I think Patrick, you're next.
Yeah, thanks, Aoife. Yeah, we just wanted to, just for 5 minutes, I think I've just been asked just to look at actually how we fund some of this biodiversity baselining. And you know, this is all information that, as probably the way that the whole farming and environmental management is being shaped towards the future is, we're all going to have to be acutely aware of the species that we have on our farm and the environmental impact that we're all having. And if we're going to monitor all that and we're going to be able to demonstrate the environmental good that we can do, we have to be at a baseline in the first place. So for me on a farm like this, it's all about how can we extract the most money from everywhere else out of our own pocket, our own pocket. And for us, the options that are in place through the stewardship scheme are very similar to some of the things that we've been doing with this AHDB trial work. So the flowering strips that we've been talking about and Aoife's just been talking about, they really are just the ABA option with the stewardship scheme, and those ABA options are worth £628 a hectare at the moment. So it's making the most of stewardship schemes as they are at the moment and as they come in the future, to actually look at all the different things that we know can benefit a farm and a farm business. So all that marginal land, all the environmental and species management, and fitting it all around good environmental schemes and well put together schemes. Then also we know private funding is coming in the future, and I think there's going to be a lot of kind of intricate head scratching from farmers and land managers when they look at whether taking money from the private sector is something they want to do. We know there's going to be a market for carbon trading, carbon offsetting. There's going to be a market for biodiversity offsetting. But I think the word of warning for that one is making sure that you know what you're selling and what you're giving away, because actually if a company wants to offset their own carbon footprint against a farm, then actually they're going to want something in return as well. If they're giving money, they're going to need something in return for that. So there's a balance for there in all those farm offices, making sure that you're making good business decisions based on a commodity that you can't actually see. Why do we want to be making sure that we do this? Well, we want to make sure that we've got really good records, we've got really good baseline data, and it is something that people can fund ourselves. It is possible to go out there and get ecologists to actually do surveys for you, assess all those different things that you've got to have that kind of that baseline data. And self-funding is another decision that farm offices might be thinking about. We've had great experiences with volunteers, and the two photographs we've got on this slide are from a volunteer photographer called Hedley Wright, who visits our farm fairly regularly. He actually came out as a dragonfly recorder. He wanted to do some photography and he wanted to do some dragonfly surveys. So we gave him a free run of the whole farm, basically in return for keeping records and giving me the records of what he saw when he was here. Then also we've got all these fantastic photographs which we have as a record, but also in all the other talks and presentations that we're able to do, we have all these photographs. So finding volunteers, there's loads and loads of volunteers out there who just want to be able to indulge in their hobbies, whether it's recording, whether it's bird watching, whether it's plant records, whether it's photography, whatever it is. Finding those people is really a skill, and there's a lot of benefit in that. And that's something, again, for a farm just to weigh up. Some farms are pretty averse to having people having a free run off site, but there is real good benefits in that as well. Also, NGOs, universities, whoever it might be who are running projects are always looking for study sites, and as farmers and as landowners we have an absolute wealth of kind of information and opportunities at our fingertips. We're involved with the University of Suffolk, who are doing lots of mammal studies. We've done some work with the Game and Wildlife Conservation Trust, who are looking at the abundance of bees in spring bean crops. And pretty much every time there's an appeal that goes out on a website or in a newsletter saying they're looking for something to study, we're all over it, because I know the resource we've got here. And all these organisations are looking for this type of thing. So sometimes it's about being welcoming, which isn't always the easiest thing to do as a farmer, and trust the people who you're letting out on your land. But there is great benefits to that as well. And the final thing is actually, it's not within the realms of impossibility to actually do a lot of these things yourself. A lot of what Aoife's been doing is trying to kind of create metrics that any farmer can do on their own land, and things like putting the solitary bee nests out is something that anyone can do, but also doing your own recording. So the Game and Wildlife Conservation Trust have a partridge count scheme, which all it really is recording partridges and getting that data into the partridge scheme, or the big farmland bird count, where you literally just have to go out and count birds. There is a lot of this that people who spend all day, every day outdoors in the countryside actually have these skills without realizing a lot of the time. And I meet a lot of farmers who say, oh, I don't really know what I've got on my farm. I say, well, tell me. They say, oh, we've got yellowhammers, we've got grey partridges, and I heard a turtle dove. And actually a lot of farmers do know the basics. They just maybe think that they need to be at a much more technical level than they are. So there is an awful lot of this that people can do themselves. And the more records we can keep and the more information you can filter out into projects, then the greater benefit it will have in the long term. Okay, that's me.
Thank you, Patrick. Thank you, everyone. A great introduction to baselining and reasons for baselining. Just a reminder to everybody, if you have any questions, please do type them into the box on the right. I think we're waiting for some more questions. Whilst we're doing that, I will jump into our next section, which is all about cover crops. So we'll be looking at effects on soil health, water quality and biodiversity of cover crops. After that, we'll come back and have a more in-depth look at the flowering strips trials. So if I'll hand over back to Nathan. Thank you.
Okay, thank you very much. And as I mentioned in my introductory slides, I will just talk a bit about what we've been doing at the Strategic Farm East in terms of using cover crops to mitigate against nitrate pollution. So next slide, please. So the main objective really of the project, or the trial, was to determine the role that cover crops have in reducing nitrate losses across the rotation. So not only just after you've grown them, but also after those following crops. There's obviously a little bit more unknown about how those cover crops may affect nutrient and nitrogen in particular in the rotation. So the trial was set up in 2 fields, split basically between areas that were treated in slightly different ways. So on the left-hand side there in apple tree, we had plough with a cover crop, and then an area where we had just an overwintered plough cultivation. And then in the other field, blacksmiths, we looked at stubble with a cover crop against stubble alone, and then looked at that, monitored that through the farm rotation. And as I mentioned, the sort of split field design allows us also to monitor the drainage water. So the drains from each of those areas were selected or used from the drainage maps to identify the field drains draining from those areas, so that we can match those management approaches to the nitrate and other quality measurements taken in the water samples. We also monitored soil mineral nitrogen supply at various times in the year to try and see what was being released into the soil, what was then being utilised by the crop, or what potentially was there at risk to leaching. Next slide, please. So as we all are very much aware, the weather has probably the biggest impact on a lot of farming activities. So this is the weather data from Brian's farm over the last 2 seasons, from September 2020 through to August 2022. And we can see that the red and the blue lines are the minimum and maximum temperature. The main thing I was focusing on really here is the precipitation. So they're the bars. So the grey bar is the actual measured on the farm. The blue is the long-term mean average of rainfall for a given month. And we can see there the variation from month to month and also year to year. Can you just press? Sorry, I've got this animated this slide. So just to highlight a few areas there. So in the autumn or winter of 2020, we had particular months there with higher than average rainfall. And if we just press again, we also identified in the spring a much lower than average rainfall, and obviously that does have impact on how that nitrate moves through the soil profile. And then finally, if we just move on again, there's one area again just to highlight differences between seasons. So the season just gone in sort of last autumn 21 and going into winter 22, we had probably one of the driest periods recorded for a very long time. And that actually, once we got to spring, persisted right through the growing season. So from March onwards, you can see there we were below the average rainfall for all the growing period. So that does have impacts, and we'll come to that. Next slide, please. So this is quite a complicated slide, but two areas I wanted just to focus on. So the first of the two top maps, sort of on the left in the centre of your screen, and this was part of a colleague who's looking at yield sort of stability and looking at how fields vary from a longer term, a cross rotation element. But it's helpful to use this information to understand how your fields vary, because obviously not all areas in the same field respond and yield in the same way, and this clearly demonstrates what we call clusters, or areas that yield very similarly. So we can see there the middle map there at the top has what we call smooth clusters, and it clearly identifies areas, for example, a headland, but also the two main areas that we were monitoring, the left and the right hand side of the field, which were actually our treatments, also had some difference in their longer term performance. And then again, if we look at the chart at the bottom of the screen, these are those zones that were, or clusters that were identified, and really just to focus in on those top two lines, which are the main areas of the field, we can see how the longer term performance of them was actually pretty similar through from 2011 to 2019. In 2019 was a winter wheat crop. Then we, after this harvest of that winter wheat crop, we put in the cover crops, and then that was followed by spring barley in 2019. Sorry, spring 2019, and then that looked at the performance of those areas. And you can note there we did get a drop in yields from where the cover crops have been, which I believe was associated with some slightly lower establishment in the spring barley after the cover crops. And obviously there are lots of challenges of how we manage those cover crops to create a good seed bed for the following crop. But just taking note, there was a reduction in yield there, which I think, as I'll go through some slides, has some implications. Next slide, please. This slide is very similar to the last slide, so I won't go into the same detail, and it basically shows a very similar pattern with areas identified with the performance, and again those two zones of the field that we'll be monitoring, with and without the cover crop, again very similar in terms of a stable yield across seasons, but a divergence when we had the cover crop, again with a lower yield in the spring barley, associated, as I say, with how that crop performed after the cover crop. And then now I'll just talk through some of the results in terms of soil mineral nitrogen captured in those crops, and then implications for how that then affected the nitrates in the water that we also monitored. So next slide, please. So this is soil mineral nitrogen monitored in the autumn and then again in the spring. So the blue bars are the autumn SMN, so that's down to 90 centimetres. This was after the winter wheat and prior to with the cover crops in the autumn. And we can see basically that the residual nitrogen after the winter wheat was very stable in all four treatments. Once we got to the spring, we can see there that the grey bars are the soil mineral nitrogen in the spring. We can see there where the cover crops have been, that you've got higher levels of mineral nitrogen actually in the soil profile. Because you've got a cover crop obviously taking up the nitrogen, you also have a certain amount of nitrogen in the biomass, which is the green bars. And obviously those two grey bars in the ploughed treatment and the stubble treatment are basically the soil mineral nitrogen in the soil profile, but because we haven't got a cover crop, you note there those are lower than the where we had the cover crop, and obviously that does potentially show that the absence of a cover crop can have a higher risk of that nitrogen leaching into the water. Next slide, please. And this is clearly seen in the water quality results from that season. So these were with drains running from about mid-November through to February. Again, very clearly matching those results from the soil mineral nitrogen. Where we had excess amounts, there was a greater risk of the nitrogen getting into the water. And we can see there, particularly the plough and the overwinter stubble areas, having quite high nitrate concentrations in the water that was above the EC drinking water level. We can see there the cover crops, one of the fields started with a higher level again where we ploughed. So again, mineralisation was likely to increase that level of nitrogen movement into the profile and potentially through the profile. But where we'd got the cover crop with the one pass system, we're obviously really low. And again, later, once that cover crop got established, we can see how that cover crop there in the ploughed field had also reduced to much lower levels. So, you know, there's very good data, and as presented earlier, showing how cover crops can scavenge, if you like, that excess nitrate and reduce the risk of losses to water. Next slide, please. So one of the challenges obviously is actually, as I say, through the rotation, where does that nitrogen that's captured by that cover crop, where does it end? Do we see benefits in terms of it being released for the following crop, or are we seeing the potential risk for it still to be leading to nitrate leaching into the water? So these, again, same charts. This is the nitrate measured in the autumn, soil mineral nitrogen measured in the autumn in each of the treatments. And again, you can see fairly stable, except for that cover crop with the one pass system where we had elevated levels of soil mineral nitrogen. And then in the spring, this was with the spring barley that was undersown with the herbage grass. So the herbage grass was just being established and starting to pick up that nitrogen that was in the soil. But obviously, it being in the first year, the roots hadn't really got as established for picking some of that up. So next slide, please. And as a result of that excess nitrogen in the system, we did see a very clear trend in the water samples, in the drainage samples, where that cover crop with the one pass system, where we had that excess nitrogen over that winter period, which, if you remember back to the weather data, indicated we had quite high levels of winter rainfall, increased that risk of leaching, and hence why we saw those spikes in the nitrate concentration in the water. Next slide, please. So following on from that, again, monitoring it through the seasons. So this is the second year of the herbage grass, obviously much more established in terms of its root system. When we look at the autumn soil mineral nitrogen, again, you can see we had much lower levels. That obviously was less risk for leaching. We also had that very different season in terms of the rainfall, so it was much drier. And obviously in the spring, again, we can see that there is a fairly uniform distribution of soil mineral nitrogen across the treatment. Next slide, please. And as a result of those much lower soil mineral nitrogen levels, we saw very much a lower risk of nitrate leaching. Now, as I say, in part of that is due to the nitrogen in the soil obviously being captured by the grass, and of course there was the dry period of weather that obviously does much reduce the risk of nitrate leaching. So next slide, please. So in summary, you know, well, I think there is very clear data now to show the benefits of capturing nitrogen between cash crops, if you like, for mitigating against nitrate leaching. However, the release of the nitrogen from those cover crops in the following crops is much harder to pinpoint, and it's quite a complicated science. There's many factors involved. Again, very much driven by climatic conditions, so the moisture, the temperature, how those bacteria mineralize the nitrogen, and the time span of that is very difficult to really give a very accurate figure on. And obviously the type of cover crops you grow and their carbon nitrogen ratio, and effectively the lignin content and how robust those residues are before they break down, will also have a big impact on the dynamics of mineralization. In addition to that, crop management, e.g. the cultivation and the cropping and the nitrogen applications rates and timings also have an influence, and we can see where we had that slightly lower yield in the spring barley following the cover crop. There was probably there was a standard amount of nitrogen applied to that spring barley crop across the fields, I suspect because it wasn't quite as well utilised in those areas that were lower yield performing, there was some excess nitrogen that increased that risk of leaching, which we then saw in the subsequent seasons. So it is very important to be able to monitor our crops through the season and try and match as best we can to the yield potential of those, and maybe make adjustments in season to the nutrition of those crops. But also, as we saw, and something we can't control, is the seasonal weather patterns and the rainfall, and that has obviously a much bigger driver on the risk of leaching. So if we get excess winter rainfall, that any nitrogen that's not being captured by root systems is likely to be at risk from leaching. So thank you very much, and I'll pass over to Fiona.
Thanks, Nathan. Yes, so I'm Fiona Burnett from SRUC, and we've been working with David Aglin, the farm manager, for the strategic farm in Scotland. So today I wanted to talk about some of our results from the cover crop trials that we've been doing on that farm. If I could have the first slide, please. Yes, so not a lot of time today for background on the farm, but located in the middle of Fife there. And the trial we were running this year was in spring barley, where we were essentially working in 2 fields where we had replicated treatments. And then we had a set of measures that were around quantifying the soil health and looking at the crop metrics in terms of how the spring barley established and how it yielded, the disease levels, and the cover, you know, the area it covered and things. But yield is only part of the story. So we were also doing measures that were around surveying the key pests, the weeds, and those natural enemies to look at some of the potential biodiversity benefits that we might get from different cover crop management methods. So if you could give me the next slide. This just shows you how the trials were laid out in the 2 fields we were working in. So essentially what we had was spring barley trials where we were looking at optimizing the sow date. With that idea, we'd have greater flexibility if we could get to the optimum sow date. In terms of the biodiversity surveys, we just picked the middle sow date for that, thinking it would be most likely the more optimal or the most typical. And then these biodiversity surveys were led by my colleague Lorna Cole, who Aoife has already referenced. So yeah, we had 2 fields set out there, and the management options, the cover crop was a mixture of forage rye, peas and beans. And we either had areas of no cover crop, we had treatments where we sprayed the cover crop off before drilling the spring barley, or we had treatments where the cover crop was retained until after drilling. So the spring barley was direct drilled into the cover crop and then it was burned off. We couldn't look at everything, so we picked sort of key pests being aphids and slugs, and then we picked some key beneficials as well. And Aoife's done a great description of the methods that we used, and it is one of the kind of strengths of the farm network that we can collaborate on methods like that. So essentially very similar, slug traps, pitfall traps, and then vegetation quadrats to count the weed species. Next slide, please. So this just shows some of the results. So here we're looking at slugs in the 2 fields, so Tank Wilson on the left there and Tal Park on the right. And then our different cover crop management options there. So no cover crop in pink, sprayed off after drilling or sprayed before drilling in blue there. And really a key point that will come out through the results that I'll present is the inherent differences between the 2 fields. So more slugs in Tal Park, just as the sort of background to that. And then also some differences in what the kind of key findings are. But I think what you see from the data here that is in terms of the 2 treatments where we had cover crops, slug numbers are significantly lower in both fields where the cover crop was retained until after drilling. Where the big difference there is the influence of having no cover crops. So we have very low slug numbers in Tank Wilson, and we have quite high numbers in Tal Park there. But potential there that delayed drilling, delayed spring off of the cover crop could reduce slug densities. Next slide, please. Aphids are one of the key species we were going to monitor, but we only had 4 aphids in the whole trial despite much scrutinising, so I would hesitate to hang very much on the findings for this year, but an observation that we only found the aphids in those later retained cover crops, so hinting at that potential risk of cover crops acting as a green bridge. But like I say, I would really hesitate to hang very much on 4 little aphids. Next slide, please. One of the key groups of species we were looking at were spiders. So obviously they are looking for species that would prey on aphids, slug eggs, and so on. So the 2 key ones that Lorna was tracking were the wolf spider and the money spider. And again, you see differences between the 2 fields. But again, that hint, that delayed spraying off of the cover crop, so retaining it until after drilling, appeared to benefit the money spiders, which are the top set of graphs there. So you see the yellow treatment there pulling ahead of the blue one where we burned the cover crop off before drilling. Again, interestingly, you have that sort of variation between the 2 fields in the effect of having no cover crop there. Wolf spiders were less common, so a bit broadly the same trend is there, but you can see that the numbers of wolf spiders are much lower. Next slide, please. We also looked at both the abundance and the diversity of ground beetles there. So again, that group of ground beetles really key in, you know, eating living and dead pest species. And diversity being important in that obviously they prey on different things, but they will also have different, you know, optimums in terms of time, temperature, moisture, and so on. So a diversity bringing potentially a bit of resilience. And again, that story about big differences in populations between the 2 fields really comes through. So you see, you know, quite significant differences in the types of species, not the types, but the proportions of species that were found. Lorna was very excited to have the sort of high abundance of the Harpalus rufipes there, which is surprising. I mean, it's normally only present in low densities in arable fields, so it was pleasing to see higher numbers in the trials here. Next slide, please. So if we start to graph that by the treatments, you can see that we have abundance in the top set of graphs there, and richness on the right-hand side, and the 2 fields sitting side by side. So again, that delayed spraying off of the cover crop until after drilling appearing to benefit ground beetle abundance and richness. So twin benefits there. And again, in terms of the story in Towle Park, that holds true where the retention of the cover crop is more beneficial even compared to the no cover crop. The no cover crop option in Tank Wilson gives you that slightly strange story where it's comparable to retaining the cover crop for a long time. So yeah, differences between fields being a really sort of key message coming out of field trial work. Next slide, please. Looking at weeds again, different weed problems between the 2 fields. Towle Park had the higher incidence of weed species, and groundsel was kind of the main species recorded there, whereas mayweed was the most frequently occurring weed in Tank Wilson. But interestingly here we have the highest weed burden in the no cover crop treatments. So, you know, the sort of impact of cover crop management there is not significantly different when it comes to weed management, but they're both lower than that no cover crop option. Next slide, please. So some of the take-home messages from this, I mean, it is only 1 year of these trials and we're repeating them again this year, but absolutely, the kind of back history of the fields appears to be really important. So we had very different predator numbers and weed assemblages between the two fields. We had that sort of fairly consistent message of delayed termination of the cover crop appearing to benefit natural enemies. And we also saw it appearing to reduce slug numbers, perhaps due to that increased biocontrol. So some sort of interesting messages coming out there that the way we manage our cover crops could be important in terms of the biodiversity and the beneficials that we see in fields. And then I've got a few slides that look at the soil structure and chemistry, so thanks. We've already had a good introduction to what VEST scores mean, so that visual assessment of how good the soils appear on visual inspections. Our numbers are slightly higher than the ones you've seen, which higher numbers are not a good thing when it comes to VEST scoring. But you do see a hint that, you know, the tile park field is ranging from moderate to good scores, but it's that middle treatment there where the spring barley was drilled into the standing cover crop is comparable to the no cover crop, but it's slightly better. It's not significant. There is a trend to it being better to where the cover crop was sprayed off early. And a hint of the same effect in Tank Wilson fields. So the one that's trending ahead there is again the cover crop being retained and the spring barley being direct drilled into it. And then the next slide, I think, begins to look at the soil chemistry. Yes. So here we have a measure of the soil nitrates. So really the May one where the crop had been fertilised is the key one to look at. Again, these differences are not significant, but the orange treatment there, which is that spring barley drilled into the standing cover crop, is where we saw the lower nitrate levels in both of the fields. So as Nathan has described there, the potential that cover crops are mopping up some of those potentially leachable nitrates. Next slide, please. And then some interesting yield results, and we described these in detail at the seminar last week if you want to look back at what was recorded. But essentially, again, slightly different messages between the two fields. So if we look at Tile Park there, the treatment at the top there, the A batch, is the different drill dates for that treatment where the cover crop was retained until after drilling. And in that treatment, you can see that there's a yield penalty, particularly in that late drilled option. So the grey bar, which is the later drilled spring barley plots. If you look at Tank Wilson, in fact, the differences between yields are not significant. And if we flip that potential challenge to having a cover crop in the field and how it impacts on spring barley establishment, I mean, Nathan, you referenced that as well. I would turn that message on its head and say that if we optimise the sow date of spring barley, we actually have that flexibility in terms of how we manage a cover crop. So picking the good news out of the bad, if you like. And then if I could flip on to the next slide, just to cover off those key findings. Interesting results from the cover crop management trials at the Strategic Farm in Scotland last year. So many trends and some definite significance to some of the data there where we retained the cover crop for the longest. So a slightly improved VEST score. That yield penalty that was evident for later drilled barley where the cover crop was retained. But like I say, I think there's a message there about if we can optimise drilling dates, we have flexibility in how we manage our cover crops. That trend to improve soil structure, lower nitrates. We also saw lower ammonium levels where the cover crop was retained longest. And then when it comes to the beneficials, that, you know, the field characteristics are certainly very important in what we see, but the timing of cover crop termination was certainly important and had a greater impact on pest and beneficials than just simply the presence or absence of a cover crop. So yes, you know, findings for this first year tend to indicate that later termination of cover crops has potential to be an important tool in supporting natural enemies and being part of those integrated pest management strategies that we're all striving for. Thank you. And I'm handing back to Callum now.
Before we go on to Callum, just had a question, Fiona, about the drilling condition. Were they challenging in Tile Park on the last drilled plots?
A good question. So we had, it wasn't as dry a spring as you suffered down south, but it was a relatively dry spring. But in that sense, it wasn't by any means the most challenging season we've had. The drilling date that we were doing the biodiversity assessments on was the 4th of April, which was a fortnight after the first drilling date that we described in the yields there. And then the latest drilling one was the 18th of April. So again, a fortnight after the ones that we were optimizing. So generally drilling date 2 was the optimum. So we were about the optimum in the biodiversity assessments that we were doing, if that helps.
Thanks, Fiona. Brilliant. Over to you, Callum.
Hello again, everyone. So I'm going to be talking about another of the projects that we've got at Strategic Farm South. So this one looks at cover crops and water quality. So next slide, please. So the structure of this project is that we have a field, and effectively at one end of it, a field's called slope field. At one end of it, we have 4 different strips, one of which was a bare stubble strip. The next was contained a cover crop mix of radish, vetch, phacelia. The next oats and mustard, and the next buckwheat, linseed, radish, and phacelia. And these are sort of 18-metre wide strips that ran the width of that part of the field. We take those cover crop strips of different cover crop mixes and compare them against a winter wheat crop in an adjacent field, sort of directly next door, called Workshop. And that winter wheat crop effectively provides a bit of a snapshot of the benefits that a winter crop provides versus those sort of cover crops in terms of retaining soil where it is and soil structure benefits and those sorts of things. So in terms of what we've done over the years, so we've again monitored things like the establishment and that sort of thing, and looked at soil mineral nitrogen and above ground biomass to look at actually where you get those differential benefits of the different mixes in the field. Next slide, please. So when we look at establishment data, some of this is actually really interesting. In some of the work packages or projects that we've got at Strategic Farm South, this sort of data is mostly used to make sure that the fields are comparable. But here you can actually use it to pick out some interesting trends. So one of the things that we noticed was that actually different cover crop mixes, different species of different cover crop mixes, take that little bit of extra time to sort of kick off and get going. So actually, early on in the season, as you'd imagine, you get things like buckwheat that actually provide really good above ground coverage and actually perform really well as a cover crop, providing that early season coverage. Whereas things like mustard take that little while longer to get going, but once they do, provide a decent chunk of the biomass within that area. So really the point of this slide is to look at actually when you're thinking about the right cover crop mix for your field site, actually one of the things to think about is when in the season you get that growth and development, and making sure that you get a good mixture of sort of through the season coverage and biomass development. Next slide, please. So we then, after undertaking some initial establishment assessments, so those initial establishment assessments, straightforward quadrats effectively, percentage ground cover within those. We then looked at biomass and broke that down by species, looked at above ground and below ground biomass in the winter of 2021. So we effectively here wanted to see how different species within the mix contribute to that below and above ground biomass to see actually where those differences are present. So I should mention that you'll notice that for the bare stubble, the value is quite high for both above and below ground biomass. That's largely because there was a lot of cereal volunteers in that area, and actually those cereal volunteers provided quite a bit of biomass in that area. So although it's bare stubble, you have to imagine that actually those volunteers will still have roots present, and they'll still actually be providing some of those same services in terms of binding that soil structure together at the surface and providing that some of that above ground cover. So some of the differences that we can pick out here are that effectively the phacelia provided really good above ground biomass, but actually in terms of the below ground, it had a bit of a reduced impact, but does provide that really good canopy. Whereas actually when you look at things like the oil radish and the mustard, those provide really good below ground biomass and really sort of bulk out that root system in terms of holding those aggregates together at the surface. So again, it's about picking mixes that actually are going to provide that balance between the above ground coverage, preventing things like capping, that sort of thing, and the below ground for retaining that soil in place, those roots to hold open biopores and provide that physical structuring. Next slide, please. So again, the next thing that we did then was we looked at soil health score. We soil health scorecarded these sites basically. Gives you a nice overview of what's taking place in the soil in terms of some of those broader metrics. So in terms of picking out some of the differences, we'll come back to the VES in a moment, but you'll notice that the VES for the strip of cover crop that contained the buckwheat, linseed, radish and phacelia, the VES score was that bit higher. Again, remember, 1 is good, 5 poor. So 3, it did have a bit of a tighter structure, but we'll come on to that in a moment. Again, pH high, but it is on an Andover sort of Hampshire chalk. So it's not that surprising, it's just something to consider really. And in terms of the other differences, again, it comes back to really not treating this as sort of a good, acceptable, bad system, but actually as this is something that I need to look at in a bit more detail. For example, actually for the potassium values, they were only narrowly either side of a threshold. That amber value isn't actually significantly lower. It's just where it's that other side of the sort of traffic light threshold. So it's important really when you produce these to look at the initial color coding to get that first snapshot and then look in a bit more data. But really, most of these values are very similar across the sites, which in many ways is good. It means that actually when we come to look at some of those other comparisons, we can make fair comparisons. Next slide, please. So again, these are those photos, those snapshots of the VES assessments that we undertook. And you can see these are rounded numbers. I also like to use a decimal point in order to just give that bit more detail. But when rounding them, you can see that actually that buckwheat, linseed, radish and phacelia score, it is that bit higher. You can see that tightly bound bit at the top. It was a little bit compacted at the surface, whereas the others have that lovely friable, sort of crumbly, stable aggregate, small aggregate texture. In terms of why that buckwheat, linseed, radish and phacelia mix was that bit tighter, that strip of cover crop was nearer to the headlands, so it's most likely an effect of being nearer to the headland, probably experiencing more traffic. I would expect it's probably a feature such as that, rather than any kind of effect of the cover crop mix. Next slide, please. So we then in November and March, either side of winter, took soil samples for soil mineral nitrogen as well. So I should add that for the future years, for this coming year, we'll also be sending off leaf tissue samples. Part of that is so that we can tie up the other end of the story. So as Nathan was explaining really well, you can look at what's in the soil and then actually directly look at what's gone into that cover crop material as well. So in terms of looking at the soil mineral nitrogen, it was lower in all of the cover crops in Slopefield versus the winter wheat in workshop. Again, by looking at the tissue samples in future years, we'll be able to tie off that other side of the story as to where that nitrogen has gone. The buckwheat, linseed, radish and phacelia mix contain the lowest of the soil mineral nitrogen values at that early November sampling time. Again, we'll be able to tie it off in the coming year in terms of making sure it has gone into the plant, and that's what we're capturing there. And actually, between that November and the March recording dates, there was a drop in soil mineral nitrogen at all of those sites. You'd imagine it's where those plants are taking up that N and storing it over that winter period. In terms of the soil water sampling for this project, my understanding is that unfortunately the sort of organisations that are managing the soil water sampling, so in each of these strips, porous pots were placed underneath the soil surface to effectively capture soil water samples. My understanding is there was a bit of a technical issue in terms of collecting sufficient water samples, so we unfortunately don't have any data on the actual soil water for this year. Trial's been relocated, and there's a hope that that's sort of should negate those issues that we had this year. We should get that data next year, so we should be able to tie off the tissue, the soil, the plant tissue, the soil, and also the soil water, and see where that ends moving. Next slide, please. Oh. Brilliant. So there we go. So so in terms of looking at then again tying it off with that with those end of year results, how have the spring crops performed? So these went into so obviously workshop remained winter wheat, and the slope cover crop mixes then went into a spring wheat. Obviously, relative to the winter wheat, the spring wheat yielded that little bit lower, but actually there weren't any significant differences in spring wheat yield between those different cover crop strips. And even when you compare it to the rest of the sloped field, sort of average across that rest of the field, there also weren't any significant differences there. So actually here, we've seen that the cover crop mixes didn't actually have a significant impact on the yield that was achievable at those different sites. Then in terms of looking at other features, such as sort of grain quality, such as the protein content, there weren't significant differences there. There was very low variability there, as you would expect between samples in the cover crop strips, but those values are very similar between different sites. So in terms of yield and actual grain quality, very similar values there. So it means that actually there's sort of two perspectives that you can have on this. One of which is the slightly cynical, why bother with cover crops if they you know, don't have any tangible effect. The other one is that actually in some instances you see that sort of deleterious effect that cover crops can have on yield. Here we didn't actually see an effect, so it's quite possible that you can use those cover crops and actually still retain that decent yield. Next slide, please. So in terms of the take-home messages, this is the first year of the multi-year project. So in terms of conclusive trends, as I've said previously, the soil data moves very slowly. So you'd expect to see those trends developing over time as you build up that time-resolved data set over the coming years. But there's a few initial kind of things to consider basically that have come out of this data. So things like when you're, so for example, considerations when you're choosing a cover crop mix to provide various services. You might want to think about things like the rate of establishment when those different species establish, the canopy development, when they develop, which species within the mix contribute to that canopy and which contribute to that root biomass, to building those root structures to hold open biopores and the like. And that's visible in the data from this project, from this first year. There's no indication also, as I say, that the cover crop mix has had a deleterious effect on the wheat crop health or yield. So really, again, it's about building up that time resolved data set, seeing how that takes place over time and whether we actually replicate the fact that we didn't see that deleterious effect in future years. Next slide, and that's me done.
Thank you, Callum. Hopefully you can still hear me. I'm having trouble putting my webcam on, but we do have a question. So I'm not sure who wants to take this one, but it's about putting the nitrogen through an animal. So grazing cover crops, do you have any experiences or views on whether that is better than grazing or than ploughing or burning the cover crop off, and how would you show the difference?
I don't have the answer to that now, but we're going to be testing that this year, or next year rather, in a replicate of this year's trials at the Strategic Farm in Scotland. So one to answer next year.
Thank you, Fiona. And in terms of another question we had was about the stubbles and whether could you comment on the comparison of stubbles and live cover crops, particularly the effect of root exudates from live cover crops compared to stubble? Is it enough to be visible in the assessments?
So I would say in terms of the stubble treatment that we had in the Strategic Farm South project, the difficulty that we had there was that obviously, as I say, we did have those volunteers still providing biomass. They would still see sort of some effects from there. In terms of exudates from roots and cover crops, I would imagine that there would be other effects taking place at a more sort of macro scale that would be more dominant factors than things like root exudates in terms of things like nutrient status and the like. I would imagine that more like actually the convective force of having roots in place, drawing those nutrients in, would have a greater sort of impact rather than something like root exudates mobilizing otherwise fixed nutrients in the soil. But happy to hear others' views on it.
Thank you, Callum. And the last question on my list for this section is it's really around looking at immediate yield versus wider benefits of cover crops. I wonder whether our strategic farm hosts have a view on that. So David Miller, David Aglin, or Brian Barker, or Patrick.
Yeah, I think yield is a difficult one to hang your hat on as being an ultimate indicator. We definitely look at margin much better than yield. Running the system we do is a much cheaper system to run. So massive savings across other areas. But I think yield is something that maybe can be a real red herring, that we're trying to produce a more sustainable and nutritious crop, which we could throw a lot more fertilizer on, etc., and get the yield back. But ultimately, the very slow process of improving soil health is a greater benefit in the longer term.
I would just add that cover crops add risk as well, because they are very good at everything else, water quality, adding biodiversity. They're very good for sort of all the insects that we'll be talking about in the later section. But yeah, it does add risk. We've had examples of the trial that we had that yield was negative, but that was more by the mistakes that I had made with pest slugs were a problem. Then after, and the season that we had, so it does add risk. I have a love-hate relationship with cover crops. They're very good for everything that we're talking about, water protection, adding everything that we need to into our soil health and everything, but they do add risk, I believe, to our following crops, and you just got to be mindful of that when you go into it, and you've got to go with your eyes open, I'd say.
Thank you, Brian. And David Aglin, did you have any views?
I'd agree with both David and Brian, actually, on both parts. It's about margin. It's also, we do see risk attached with it. You could see clearly in our results last week that there was a yield dip as a result of keeping cover crops longer. But the benefits for us as a business are big enough to not necessarily put up with that in the long term, but I think we can learn how to mitigate that, and that's the idea of looking at sheep grazing next year to see if we can return some of that fertility and ultimately maintain hopefully the yield of the following crop, as well as getting all the physical benefits that Brian mentioned of the cover crops, so we get a win-win hopefully.
Thank you, David. Good. Okay. Well, we are slightly overrunning, so let's look at any remaining questions right at the end. But now let's have a look at the flowering strips trial. So if Aoife and Patrick could join us. Thank you very much.
You're lucky I talk quite fast, Anna, so we can get through this in 10 minutes. Okay, so we're going to close out today looking at the flowering strips work at Brian and Patrick's in Suffolk. So just get the next slide, please. So earlier on, we did a very quick run through on the baselining that we did and the different monitoring methods that we used. And again, in the next couple of slides, we're just going to do a very high-level summary of the insects, the plant species that we found, and also the economics of the flowering strips as well. But I just wanted to start by kind of just showing you what the flowering margins actually looked like throughout the seasons. So we had 2 fields with flowering strips sown in them, and 1 field without. And this is when we first went to Brian and Patrick's in November in 2020. And so what we had was there in the flowering strips that were around the outside of the field, they were sown in 2019. And when we went in November 2020, they had been a year established. So you can see when we first went, this is what it looked like. And if you, I guess what I'm trying to show here is that when you look straight down the margin, you just think it looks like, you know, a field of green, but actually when you look down and the diversity of the species that are in there, you've probably got 12, 13, 14 different kind of flowering and different perennial species in there just in November alone. And then you move further through the season and that same flowering strip. So what it looks like as you go into April, May and June. And so the bottom picture there is the oxeye daisy. This is the flower that normally comes in first, and it tends to take over the margins in the first year of sowing. And then you move further into the season and you get different flowers emerging. And what we looked at was the change in flowering species over time, but also within the margins itself and then across fields as well. And so I've just got some pictures there of the quadrats that we took, and you can see just in one quadrat alone, the variation there in the number of plant species. We were kind of getting, you know, 13, 14 different grass and flowering species at any one time. So a huge diversity there in the species mix. I will say that the mix that was sown at the farm was an Morsgate mix. So this was an ESG1 and ESG2 mix, which are kind of the gold standard really of the flowering mixes. And so they contain a lot of finer grasses that allow for the flowers to kind of to blossom within the mixes. And there was also a bit of EMF5, which is a wildflower mix for loamy soils. So the Morsgate mixes are the gold standard, but they're also very expensive. And we'll come onto the economics a bit later. And you can just see how that compares to other mixes as well. And so then the last picture here is kind of what you're aiming for with this. So again, this is November 2021, a year after we first went, and we had a lot of debate about what to do with the margins. So should we cut it? Should we leave it? Should we just take a bit out? And we kind of came to an agreement that we would kind of leave half and cut back half. And it was baled earlier in the season. But this is basically what you're aiming for. This is kind of the ideal for wildlife habitats. So on the top, you can see that there's hedging. So the very, very high hedging there. And then we've left a bit of margin. And then you also have the grass. So you've actually got 3 habitats going on there, which will benefit lots of different wildlife that are on Brian and Patrick's farm. So yeah, this is just what the outside margins look like. And then if you move on to the next slide, this is the center margin. So the center margin was sown in summer 2020. So when we went in November 2020, it was a little bit behind the other outside margins. But again, you can see, you know, 6-7 months later, it was just beautiful, in bloom, really bright green in color with lots of flowers present. And then you move into September 2021, so three months later. And what we did with this strip was well, Brian and Patrick cut the outside of the margin. So it's a 6-meter wide margin, cut the outside and left a strip in the middle for birds and insects and other wildlife to nest in. And this is just a photo of my hand there, and I literally 30 seconds within the margin. And I think I counted like 19 different grass and flower species. So within the course of kind of less than a year, you know, it established really well, and there was a huge array of diversity in those margins. So next slide, please. Okay, so if you're thinking about using flowering margins or grass margins for to encourage natural enemies for pest control, I think the first question that you have to ask is, what are you actually trying to manage? So Fiona mentioned earlier that they were also looking at slugs and aphids, and we were doing the same. Now, similar to Fiona, we had across-field differences also in slugs. So one of the fields, Big Guinea Row, had much higher levels of slugs than the other 2 fields. But also, like Fiona, we had very few aphids. So I think if you're thinking about what mixes to use, you have to consider the pests that you're actually trying to target, if that's what you're using the margins for. Now at Brian and Patrick's, they have a lot of grass in their rotation. So one of the things they struggle with is fruit fly damage. So for fruit fly, because it's you've got the larvae in the soil, you might want to go for more kind of beetle banks and kind of tussocky grasses to encourage beetles, which will then go for the soil-borne pests. But it's something you really need to consider if you are going to be using these margins for pest control. Is what's the insect that you're trying to target? So do you have a specific problem in a field or on the farm as a whole? Is it soil-borne? Is it aerial? So if you have aphid problems, for example, across years, then maybe you want to look at something more flower-rich that will encourage things like hoverflies and ladybird larvae. If it's more soil-borne pests that you're trying to target, then you might want to go for things that will eat the soil-borne pests or the slugs, like beetles and other insects. Okay, next slide, please. So we talked earlier about the different baselining methods that we used, and the pitfall traps and the water traps. And I think what came out of the monitoring from the last 2 years was when you get your insect traps and you're not very au fait with how to do the assessments, you get a trap and you look down and you've got hundreds of different insects in that trap. Now, what do you do with those, and where do you start with that analysis? So I always say I'm not an entomologist, I'm a pathologist by training. So when I first saw these, I was getting panicked about how I was going to start it. And I think over the last 2 years, what I've learned is, if you could just click again, because I've got some animations, Christian. Start quite big. So when you tip out your pitfall traps, is just to separate them into the big groups. So we all know what spiders look like, we all know what beetles look like, we should know what things like wasps and bees and midges look like. So just separate them into those groups first, and get to know one specific group. So maybe you find spiders really interesting, or you find beetles really interesting, and just get to know those really, really well. And then just another click again, Christian. And then if you are particularly interested in beetles or spiders, then you can kind of divulge down further into those individual species. So again, like Fiona mentioned earlier, the wolf spiders, the money spiders are different beetle species. The middle guys there, so these are things like symphilids and midges. They're quite difficult to ID down to kind of very specific levels, and you really need a hand lens and kind of tweezers and stuff to look at them. So if you're not like a big enthusiast, then the bigger guys are much easier to spend time on. But I would definitely advise, don't get too bogged down in identifying individual species. Just get to know a particular group and the kind of the rough amounts of different things across fields and across the farm, because you can get very bogged down in this if you get too many insects in your traps. So the next slide. So Fiona kind of looked at some of the quantitative measurements, and just because of time today, I'm not going to go into the amounts that we found across the field, and just take a look at some of the individual species that we found. So in the last 2 years, so every year I think we've counted around 400 pitfall traps and water traps, and it's kind of roughly been anywhere between 50 and 200 insects per trap. So huge amounts of diversity across the 3 fields and across the whole farm, to be honest. And these were just some of the different insect species that we found. So common ones are things like the soil pest complex, so springtails and millipedes, ladybirds and hoverfly larvae. Again, we didn't find a lot of these, and I think that's because at the farm they don't really have an aphid problem. We struggle to find any aphids in the farm, and so the things that eat the aphids weren't very common. But we did find a lot of things like midges, and then a lot of beetles, so the generalist beetles like nebrias and the trechus, a couple of maras, and then the harpalus that Fiona mentioned as well. And then huge diversity in the spider numbers as well, both the ones that live on the crop, so money spiders, and they disperse by floating on air currents, so they use their webs to disperse that way, but also the ground dwelling ones, and they are active predators in the soil surface. So again, if you've got something like a soil-borne pest problem, like a fruit fly or wheat stem borer, then those kind of ground dwelling spiders and your generalist predators are going to be really important to manage those soil-borne pests. So next slide, please. Okay, so looking at the plant species counts within the margins. So yeah, I talked a little bit about this at the very beginning. So what we did was we took two sides of the margin, so the left-hand side and the top side, and we just did 9 quadrats, kind of 5 meters apart throughout the margin, and put the quadrat down and just did a count. We weren't looking at it, how many daisies were in the quadrat, just a general count of what species were in there. And we did those half a meter and then 5 meters in from the margin edge as well, to look at, did any of the grass or the floral species cause a weed burden. So just a couple of animations there, Christian. So the first thing you notice is the ryegrass. Now, I mentioned earlier that Brian and Patrick have a lot of grass in their rotation. So it shouldn't be too surprising that we found a lot of ryegrass in each of the quadrats. And then just 2 more animations there, Christian, so you can see, there you go. The other thing I wanted to point out as well was a couple of blackgrass plants there, but also the variation within the fields. So so bottom 59 is one field where we had the margin on the outside and then the center strip as well. And then Big Guinea Row is the other field, which is about I guess a mile and a half away. And first of all, you've got variation within the edges themselves, the 2 edges that we monitored, as well as the center margin, but also variation across the 2 fields that we looked at. So things like chewing fescue there, we found in kind of 2 of the quadrats for bottom 59, but then in none of the others. So yeah, the kind of take-home message really is the variation there, both across the field itself and within fields, and also just the diversity there of the different grass species that were found. Some of them are a natural weed problem, like your bromes and your blackgrass and ryegrass, but some of them will be really important for things like beetles and spiders as habitats. So next slide, please. And again, so these are just the different flowering species that we found at the same time. So you've got your 2 fields there, bottom 59 and Big Guinea Row, and the 3 edges that we monitored. So the edge 1 and edge 2, and then in bottom 59, the center strip. And there's just another animation there, Christian. So just looking at the kind of top 4, and these will be present on most farms, of the knapweeds. So the knapweeds come into flower, they're the first thing to come into flower normally, as well as the oxeye daisies. And then wild carrot and I guess sometimes ladies' bedstraw, they will come into flower a little bit later. So the whole point is that you have I guess kind of consistent floral resource, at least from March all the way up to October, November. But if you can manage to get something to overwinter as well, and that's not impossible, you can always find a few flowers lurking. But there will be lots of other plant species to support biodiversity throughout the season. So you have your kind of common floral species there at the top, but then there's a couple more animations there, Christian. So some unusual species there that we found, things like the smooth hawksbeard, bladder campion, false oatgrass. So you kind of get them not across all of the margins, but they are there present. And there are management options that you can use. So for example, with the oxeye daisy, that tends to be the dominant species in the first year. You can manage the margins appropriately to try and kind of reduce the numbers of oxeye daisy and encourage other flowers to kind of progress over the years. Things like bladder campion and stuff that were quite rare, there might be ways of encouraging those across the years. Okay, so next slide. So this, I suppose, is probably the most interesting bit for me from the point of view of the weed burden. So we wanted to see whether the grass and flower species that were sown found their way into the main crop. Now, you wouldn't expect that necessarily, because the flowers that were sown were all perennials, so they're not annuals, and so you wouldn't expect them to cause a burden, not in the first couple of years anyway. So I've just got some pictures there of what the space, so the sterile strips in between the margins and the crop actually look like. Now, some people have said, oh, why did you leave a sterile strip? You know, that's just a waste of space. And that's a completely valid argument. Some people like to use a sterile strip. I don't think anyone's wrong or right in that. I think it's a personal choice about what you want to do. But they do come with their own kind of complications. And I guess that's what I'm trying to get across there in the pictures. Is if you look at 2020 and 2021 with the so the top photos there are from the center margin and then one of the outside margins, and the sterile strip there looks really nice and clean in kind of 2020 at the center margin, and then in the right hand side we've got a couple of blackgrass plants, but again, you know, you can pull those out. They're quite easy to manage. The ones that you do get kind of starting to establish in that area, you can, things like groundsel and stuff, they're quite easy to pull out and keep a handle on. But the problem then came in 2022, where you get the sterile strip full of different grass species. So it does beg the question of, well, should I have left a sterile strip in the first place? How do I manage that? And yeah, there's no right or wrong answer. It's a bigger question that I don't think we can answer in this particular webinar, but it's definitely something to consider. And then just looking at the numbers, bottom 59 and Big Guinea Row, for what we found 5 meters into the margin edge. So yeah, ryegrass has always been there, a couple of blackgrass plants, and groundsel is another one. So they have started to find their way in and create a weed problem, whether that's due to the grass margins or the grass and floor margins themselves, or whether they would have been there before. I should probably leave Brian and Patrick to answer that one. But that's where we are with that, and we've got another year left of looking at this, so it'll be interesting to see how this follows on and whether we see anything going into next year. Okay, so next slide, please. Okay, cool. I won't spend very long because I can see Anna's trying to rush me through. So this is just looking at the cost of establishment. So I mentioned earlier that the Amoriscape mixes they're the gold standard. They're very, very expensive, and there are other mixes available which you can use, and that are a lot cheaper, especially if you already have margins on your farm already. But yeah, this is kind of the rough costs of establishment of the mixes. And then the next slide, please, Christian. We also looked at yield and margin across the rotation, and again, this is all in the report, and just email me if you're interested in getting this. So we calculated the loss of income for where we had the flowering strip down the centre of the field. So it's 6 meters wide, 445 meters long, and we had the yield and margin for the last 12 years. So I guess the main take home there is the increase in the margin for this year, largely due to the wheat price, so £265 a tonne, and what that loss of income increased across the rotation, and for wheat only in 2022. So that's what we have. And then the next slide, please. Yeah, so just the last point what I wanted to make was, you know, this is really nice looking. It's, you know, really well sown margin with high quality seed, you know, really expensive, and it's a really good resource for biodiversity. But not every margin has to look like that. It can also look like the next slide, please, Christian. It can also look like this. So it can be a really natural margin that's just been left to kind of grow wild, and this would support huge amounts of diversity. But then it can also look like the next slide, this, where you have floor resources within the crop itself. So is this, people will look at this very differently. Is it a weed or is it something else? And I guess that's going to be an interesting take home for the future. So I've taken these from a farm in the Cotswolds, Ed Norton, and he is very into kind of within field biodiversity. So do you see this as a weed, or do you see it as a resource for biodiversity? I think that's something to consider. Okay, that's me done. We're running over time.
Thank you, Aoife. A few questions have come in, and one of them is, how did you prepare the seed bed, and what type of drill did you use with the establishment for the flowering strips?
Patrick?
Yeah, I was hoping Brian was going to jump in there.
Oh, is Brian there?
Yeah, so establishment, we did normal stale seed bed, and then we just broadcast the seed with the grass seeding unit we've got. So it's basically just broadcast and rolled, so nothing more than that. So, and that was done, some was done in the spring, some was done in the autumn. So we sort of had good establishment on both accounts. But yeah, it was quite nerve-wracking when you're putting that expensive seed through a drill. So I actually double half the rate and did it twice.
Thank you. All right, good. We'll spend a couple of minutes with Patrick, and then we'll follow up with some final questions. Thank you, Patrick.
Yeah, thanks, Anna. Yeah, just wanted to, just for a couple of minutes, just talk about, kind of links back into what I was talking about earlier, is the different funding streams. And whilst Aoife and her team have been looking at all this from the cross crop pests and beneficials point of view, I wanted to have a really good look at all the other benefits and all the other wildlife benefits you can get from putting these mixes in. So I dreamt up my own project and went out to the Felix Thornley Cobbold Trust and got funding to fund a couple of ecologists to visit the farm over the course of the year. And you know, this is just one example of actually how we can go out there and source funding. And I basically just set them the remit of just find everything else that's using these flowering margins on the 3 different fields, and give us a real snapshot of the wildlife that we're able to attract just by putting flowering margins into around the edges of fields. A lot of the data hasn't actually come back yet, and one of the most useful things that actually happened was the ecologist I was using, who was a friend of mine, actually had COVID in one of the weeks, so he freed up 1 week's worth of funding. So I was able to get someone else in who was a specific bee and wasp specialist to give me a whole different kind of variety of what was there as well. Excuse me. So, and then linking up with other projects as well. So the Felix Thornley Trust funded the studies. The Suffolk Naturalist Society bought me moth traps, so we were able to go to study the moths that were using them. And the University of Suffolk lent me all of their mammal trail cams and their bat recording equipment. Now the mammal cameras showed all the different species you can see along the bottom there. Chinese water deer is a relatively recent addition to the farm, but what it did show was that pretty much every camera had hares just literally staring at the camera for long periods, and the hares seem to really like trail cams as well as flowering margins, as it turns out. The bat detectors and the bat data is going to be fascinating, but those results haven't come back yet. And some of the real provisional stuff that I've got back from looking at this is that actually on this farm we've recorded 25 species of butterfly over in the last kind of 15 years, and we recorded 20 species of butterfly just on the 3 flowering strips. We also recorded dragonflies hawking up and down them as well, because where you've got small flying insects, you're going to be having all these other insects that feed on those smaller insects. And partly that's why I'm so interested to see the bat data as well, because if you bring in a food source, then you're going to bring in these other species that rely on them. Having the ecologists walking about was really useful, because it meant that they found a couple of meadowpipit nests. Meadowpipits aren't a bird that we've ever known to breed on this farm before, and they found probably the same pair had a first brood and a second brood nesting actually in one of these flowering strips. And reed buntings are a species of bird that historically will always overwinter here. They'll use our areas of wild bird seed mix, but they will then disappear off. And ringing data from this farm and other places shows that the reed buntings that we've ringed in the winter here have gone to places like Lackford Lakes or to other places which are much more reed-based landscape. But we actually found that there was reed bunting nesting here as well, and they were using the flowering strips to feed their young, which was really pleasing. The bottom picture on there as well, because we'd speak to farmers a lot, I always make sure that we look at the cost of this. and over the day of haymaking, so the AB8 options and these flowering strips, we actually made 44 bales of hay at £7 each. So there was a bit of an income on there from the hay as well, which is pretty handy. And if I'm ever trying to sell it to farmers, we're always trying to make sure there's a financial benefit in there as well. Now, the chap who came out, who was the bee and wasp specialist, on the next slide, please, Christian, he actually found that the bee species were kind of far greater than he was expecting, and over the course of the day that he spent looking at these 3 flowering margins, and then also when I was talking about self-funding earlier, I paid him for a separate day to have a look at our AB1, AB9 plots and some of our other wildflower meadow areas. So I actually got 2 days of recording from him, and the vast majority of what he found was in all of these flowering strips. But he found 20 different species of bee over the 2 days that he was here, 4 species of wasp, and actually 17 species of hoverfly. Now, if you know your Eristalis tenax from your Eristalis something else, intricaria, then you'll know that hoverflies, there's hundreds, like 270 species in the UK. If, like me, to start with, that you didn't even realise there was more than one, then the fact that there was 17 was, I found that fascinating. And just having that expert wandering around looking at all these other benefits of these flowering strips and the detail that we were looking at has kind of given me a bit more lit a fire to go and find out actually what is more. And I'll be looking at some of those different funding streams to see what we can find in these areas as well. This is just some of the species that he found on here. The top 4 species are all bees. The bottom line, the first 3 are hoverflies, and the bottom right is a 6-belted clearwing, which is a moth. To the untrained eye, they all do look fairly similar, but it's amazing once you start getting into these flowering strips in these areas and you just see the diversity that's in there. The more time that you spend just looking, and the more you actually see. So I think the take-home message for me is not only is this good for our crop pest management, but it's actually brilliant just for that farmland wildlife and the whole farmland ecosystem. The more variety of habitats we have, the more farmland wildlife that you can support on your farm. And the more farmland wildlife we can support, the better our countryside is as a result. So yeah, all good stuff, all interesting stuff, and lots of good projects that all link together. Lots of hard work by lots of people.
Thank you very much, Patrick. That's brilliant, and hopefully inspires all of us to look a little bit closer when we're going around farms. So I think we'll spend just 10 minutes going over any final questions, as we've still got a lot of people watching. So I think most of the questions I've got now are on flowering strips and on biodiversity. One of them is looking at combinations of field boundaries and whether that made a difference in terms of looking at combining flowering strips with field margins, hedgerows, or short grass strips, and how that potentially impacts the biodiversity and the effect on some beneficials.
I mean, I can make a comment, Patrick, and then you can jump in. So I guess with the 3 fields that we were looking at, so bottom 59 and top 59, they're linked, and they have a hedgerow between them. And top 59 didn't have a flowering margin. So I guess it was, yeah, you could consider some linkages there about the impact of a flowering margin in one field and then none in the next. But Big Guinea Row was 1.5 miles away, so again, it's difficult to make any linkages there across the 2 fields. Don't know whether you've got anything else to add there on that, Patrick.
Yeah, on a farm like this, we always talk about the whole farm ecosystem. So the more habitats that you're able to link together, the more all these species are going to be able to benefit. So where we're putting in flowering strips, we're looking for that whole variety of insect life, because we know that they're going to be feeding on crop pests, but also at the same time, there's other species in there that want to be feeding on all the insects that actually we're trying to encourage. So where we've got flowering strips next to good thick hedgerows, we know that's really good grey partridge nesting habitat, and those grey partridge chicks want to be eating all those areas, those insects. One of the interesting things that we actually found from the bee walks was that on top 59, where there's no flowering strip, the only insects he found on that field were on a couple of patches of thistle that obviously escaped the boom of the sprayer. And other than that, the cereal crops at a time when the hedgerows had finished flowering were very light on insect life.
That's excellent. Thank you. A couple more questions came in about the costs and benefits, and whether we've included the cost of topping, because obviously the establishment of flowering strips is potentially a lot of topping cutting in the first year especially. And the other question was whether we included any AB8 stewardship payments in the margins.
So no, those weren't included in the costings. And yeah, topping is it's a very topical subject. Topping is topical. And yeah, because some people don't have balers, you know, they might have flails. So how you manage them is very, very topical. Now, speaking to a couple of different people, so Roger Draycott, for example, the GWCT, he recommended leaving the flowering strips the whole way through the season and not cutting them at all, and actually filleting them in January when they're quite crispy and hard, and it makes it quite easier. But yeah, it is down to kind of personal choice and the kit that you have, and how you manage those. And yeah, no, we didn't include the topping cost in there, and we can do that going forward. And then no, we didn't include the stewardship costs in there either. And yeah, we'll definitely look at that next year.
Thank you.
Yeah, I think the biggest factor really was the weather, because we looked at that central strip that we left down the central strip down the central strip, and actually this year the bit that we didn't cut and remove anything, the bit we left actually flowered a lot better than the bit we had cut and removed, and that was only because it was so dry, and that was just a bit of an anomaly. But when the weather's so unpredictable, there are things that are always kind of, the curveballs are sent to try and fool you a little bit. There's an awful lot of people who try and create really good wildflower habitat without the ability to cut and remove, and that's kind of the age-old quandary for flowers, is actually how do we cut and remove? Because mulching, if you have the capability of doing it, as Aoife's just said, then you can really kind of mash it up into quite a fine dust. But with old flower mowers, then all you actually do is you leave the mulch to smother the surface, and then those flowers can't push through. Something that we found on one of the areas on the Big Guinea Rose strip, where we cut that hedge this winter, and then it didn't rain. Actually, the ground was pretty hard, and the flowers couldn't push through there as well, because we've got a real kind of compact crust on the top. So there's a whole number of different factors that kind of contribute to flowers and how well they show through the summer. The things that we do during the winter has a big effect on that.
Thank you, Patrick. The next question was around the effectiveness of flowering strips for water and runoff and erosion, the leaching of nitrogen, that sort of thing. So effective, I guess, of flowering strips as opposed to perhaps more grass-based strips.
Yeah, I mean, the thing to say with those margins is they do actually have quite a lot of grasses in there. So it does have a combination of two. I'm not sure whether anyone has any comments on things like rooting and effects on soil structure. That's not something we looked at.
Okay, thank you. And then the last question I've got was about the ergot and potential for ergot to move from grass in strips into adjacent crop. Have we seen that, and do we have any advice?
Well, so ergot is a very topical subject this year, because there's been quite a lot of it around, and it is definitely a concern. So myself and one of my colleagues, John Cussons, he's a weed biologist. We're definitely concerned about the plant health effects of some of these mixes. From the point of view of, we're getting them imported, so we don't really know what's in them sometimes. But yeah, we also have to think about the knock-on effects on things like ergot, and different grasses coming into flower at different times, and then the knock-on effects of insects in those flowering grass margins, transferring the ergot into the crop itself. So yeah, it's definitely a concern, and it's something we are looking at and are looking at going forwards, definitely.
Good, thank you very much, Aoife. I think that's everything we have by way of questions, so we will just wrap up now. The next webinar will be at the same time on Wednesday, the 23rd of November, and we'll be covering tailoring crop inputs. And the other thing I just wanted to highlight is that we are looking for our next strategic farm. It's a 6-year program, and if you'd like to know more, please get in touch, and the application form is on our website. Thank you very much to all of our speakers and hosts. And thank you for everybody who joined the webinar, and we'll see you soon. Bye-bye.